Good evening, sir and madam. You all are welcome in this Science Dialogue series. As you all know that this is free webinar series we have initiated to increase knowledge pool of bioscience. In this series, as you all know that we invite very renowned people, renowned person from biology stream from different all over the world, you can say, not limited to Gujarat or India. Our resource persons are from different institute, renowned reputed institute. In this series, as you all know from the flyer, today's speaker is Dr. GIFTSON Senapathy, and we have also spent 2-3 years as a colleague in Uka University. He did his PhD from Karunya University, and then he was teaching there in some university, and then he joined as assistant professor at Uka University. Then he went again for postdoc fellowship at Johansson University. I'm pronouncing correctly, na?
Yeah,
Johannes University as a postdoc. He has also supervised several PhD students. 2-3 students have just completed PhD. Nowadays, you all know from flyer that he is working as senior assistant professor at PDEU, Pandit Deendayal Energy University. He is in Department of Biotechnology. With this, and no need to say publication and review paper, all these things, because everybody have those who are in this line, research paper, review paper. So I welcome Dr. GIFTSON in the Science Dialogue series. You are welcome. And we are grateful that you have accepted our invitation for delivering lecture, because delivering lecture is not easy, and online, always I speak this thing, because delivering online lecture in which you are not knowing the standard of the participants. So that is very daring task. If you are confident, resource person are confident, then only they can deliver. So really, I'm thankful to you for this acceptance and ready to deliver lecture. Now platform is yours.
yeah. Thank you so much, sir, for inviting me for the Science Dialogue series. I do agree, because we are in the common forum, and I could see that he organizes it almost every week, I guess, right? Almost every week, and he's inside Gujarat and outside Gujarat. I think he's well known. If you go to GSBTM, if you go to different areas, any government bodies as well, Dr. Gopal is very well known, particularly for the Science Dialogue series and many other stuff, and some of the important contributions that he's making as a professor and as a facilitator and as a spokesperson for biotechnology and biological sciences. Very few people you can see like this. There are some very good researchers, and there are some very good professors, and there are very good spokesperson, but it is very difficult to see all these people together in one, and that's what the contribution. So he's an inspiration for many people, and I do agree. And we shared the same department, and we had a lot of discussions, many other stuff, and he's very active in conducting many other training sessions for the students. So I'm thrilled. I really am amazed, and I'm thanking you again for this invitation. Without any further ado, we'll go forward to the topic, because I think most of you are waiting for it, and we have limited time. Let's have this, and after that we'll have a discussion about whatever related to the topic or anything else which you want to ask me as further. Okay, so I'm going to talk about applications of photodynamic therapy and nanomedicine in cancer therapy. So before that, let me give a very brief introduction about the background where I'm coming from. I'm working in PDU, Pandit Deendayal Energy University. Earlier it was Pandit Deendayal Petroleum University, but 2 years back, I think our Honorable Prime Minister, when he was addressing for the convocation, he wanted a petroleum university to convert into energy university, because there is a lot of need for energy, research in energy, contribution in energy for the whole of India. So that is why actually it has been converted into Pandit Deendayal Energy University. And in Pandit Deendayal Energy University, Department of Biotechnology is a very new field. You might be wondering that what biotechnology is doing in the energy university. So we are focusing on bioenergy, biomedical sciences, and many other stuff. Here, here we are mainly focused on, taking the research from the lab to the commercial setup to the end user. That is what the main thing is. So we are offering BTech Biotechnology. It is very new, and we don't have MTech, but we do have PhD as well. And we are having very well infrastructure, lab infrastructure. We are even planning to bring BSL 3 facilities. I'm working here. So if there is anyone who is visiting here, I'm ready to help you and we can work it out, right? And so this is my research background. I started my research on dietary phytochemicals, particularly on cancer therapy. Then I shifted my focus to phytotherapy because India is a hub. Our culinary cuisine is a hub of we have lot of chemicals. We have lot of stuff that is infused in our diets, which the people in abroad they don't have it. So that's why Indian cuisine, Indian diet is so famous in case if you travel abroad, not for its taste, but also for its important ingredient. Say, for example, turmeric we use every day. Turmeric we use even in milk, even in chai, even in everything. But for them, turmeric is a medicine, but we use it like anything. Like with pepper, ginger, so many. If you name the spices, everything is there. The masalas have everything, and everything has. If you have time, go and study individually. Everything has a very good, biomedical applications. Particularly, it is having rich antioxidants, and they can be used as anti-cancer agents. So, lot of research has been done. Now, the second phase of dietary phytochemicals have been started, like how we can make these dietary phytochemicals into a nanomedicine, into a nanoparticle, and how we can engineer it and how we can make it into nano robots. All right. So it's like, engineering is something. Say, for example, a simple terms in simple terms, if I want you to explain, if I want to explain you about engineering, it's nothing but when the wood is there in the tree, it is a wood. But if you cut down the wood and if you to shape it into chair or table or any other equipments or any other fancy items that you can keep it in the table, that's what you call it as engineering. Engineering is you don't think that it's something related to it's a very fashionable term. Nothing. If you change the nature and if you make some differences and if you can work on the nature and if you can modify it into a different format where it can be used for different applications, that's called as engineering. So you can engineer the phytochemicals and you can make them into nano conjugates, and that's what and the researchers. And after that, we are focusing on targeted therapy, because mostly as we go across we can talk about it. In cancer, what will happen most of the time, particularly in cancer, the treatment induced side effects will be very lethal. Say, for example, a patient who is not treated by cancer, he may live up to 14 years, but chances are there of a same person, if it is treated, he's treated by for cancer, the cancer would have reduced, the tumor would have been reduced, but the side effects will be more. His heart will be affected, kidney will be affected, the entire system will be affected, hair loss will be there. So physically and emotionally, the person will be affected. So because of the treatment induced side effects, not only the cancer is killed, but other cells also damage. So that will be, very difficult for a person to lead a life. So targeted therapy is very important. What is a targeted therapy? Instead of killing the normal cell, if you target, if the cancer is in breast, if the cancer is in lung, the medicine will go only affect the affected cells. The medicines will go and kill only the affected cells and it will spare the normal cells. So when it is sparing the normal cells, the treatment induced side effects, cancer treatment induced side effects, many times maybe you would have seen that in YouTube videos or in some ads, that people who are undergoing chemotherapy, their hair will not be there. Even for little children, the hair will not be there. That is all side effects. So when you focus on targeted therapy, these side effects will be nullified. That is what one area we are working. What are the different targets that the cancer cells are having, and how we can modify the targets, and how we can give precision medicine for it. Then one such area that I'm working all in addition to that is photodynamic therapy,
This bottom
which
line,
will be,
hide that
sorry,
line.
yeah, this one, right?
Yes, yes. Just click on hide. Yes.
Yeah. Thank you. If there is anything else that you can't hear, or if there is any disturbance in slide, please
No,
you
you're very
let me
audible and very smoothly. You're very good orator, so we know.
know. Thank you, sir. So now we are focusing on photodynamic therapy. I'll be explaining you what is photodynamic therapy in next few slides. Photo, as you know very well, that it is light. So with the help of light, how cancer can be treated. That's what the focus is. So though there are different aspects that you are working on, different strategies that you are working on, particularly on cancer, in today's talk, I'll be focusing only on these two, photodynamic therapy and the nanomedicine. How these two individually they can have an effect on cancer, and at the same time how these two, when they club together, they can have a very good effect on cancer. That is what we'll be working on. That is what we'll be seeing on, right? And I'll be showing you some results of our lab which we have done, and also some you know uh, works we have done. I'll be just telling it orally because uh, without it is getting published, I can't publish the show you the results. But few things we can discuss it orally. I can give light to you, but it will be giving you a light on what is photodynamic therapy and what is nanomedicine and everything, right? Okay, let me start with one famous quote: "You'll never win if you never begin." It's from Helen Roland. No matter whatever the presentation I give, I will start with this because it has inspired me. Most of the things, most of the things what we do is that our plans will be plenty, but our results will be empty. You'll be having a lot of plans. If you ask any young person, any young man or woman, they'll be having a lot of dreams, but not even a single step they would have pursued for the dream. So the most important thing is beginning. Okay, a well beginning is half done. That is what it. So try to begin, try to start it, whatever you think, it'll all will fall in line, right? So these are some of the projects which we have done, which we have completed. We'll just see that few. Before getting into the nanomedicine, before getting into the cancer therapy and so on, just we'll have a small idea for a few minutes, maybe for 5-10 minutes, we'll have a small idea about what exactly is cancer and why it is lethal, why people shock still they hear the word cancer. What is so difficult and what is so different about this particular disease? For that to understand, we need to understand that we are very complex human beings. In case if we think around how many atoms that we are made of, we all are chemically made, right? In case if we are thinking of how many atoms that we are made on, we can say that yeah, okay, can
Maithil, I'm removing you. Please continue now. I have removed.
okay, yeah, thank you. So this many number of atoms, if you can count the number of zeros, it will be 27. This is just an approximation, but still it will go on further as well. So 7 octillion, this number is nothing but octillion, 10 followed by 27 zeros. So we are made up of 7 octillion atoms, and you need to understand that if you can try it, if you can read it along with me before you finish reading this statement, more than 2 billion new cells would have been formed in your body. In case if you have read the statement with me, now imagine that your body has produced more than 2 billion new cells. That means you see how much our body is producing. Every minute or every second, 25 millions, approximately 25 millions new cells are formed. Now you see what is a cancer. When one cell, when it is dividing, if one cell is disturbed, if the genetics of one cell, if the mutation in one cell, if it happens, that will be carried forward, and that can lead up to cancer. Cancer is nothing but abnormal cell division. So when a body is producing this many number of new cells, why we are not growing like a Hulk? Why we are still staying on the same size? Why we are still staying on the same height or size? Why we are not growing like a different monster or something? You need to understand, though there is enormous cell multiplication, but our body is having the cell multiplication is in control. Though 25 million new cells are formed each second, it is always controlled. Now, the point is, the body or each cell has a controlling mechanism to control the cell division. When any of the genes which is working for the control of the cell division is breached, or if they are disturbed, if they are mutated, then the cell division becomes uncontrolled, and that is what you call it as cancer. Okay, so cancer in fact is nothing but uncontrolled growth of abnormal cells. If you see the statistics, 17 million new cases were there, and if it is estimated, more than 240 in 2040, more than around 28 million new cases of cancer will be there for every year around the year. You and me will be knowing, if I ask you how many types of cancer you and me will be telling up to, like, 3 4 cancers, like lung cancer, breast, colon cancer. But if you see, there are more than 150 types of cancer, and most of the cancer, out of 150 types of cancer, if you see, only 5 or 6 cancers are very lethal. That means WHO has branded lung cancer, colon cancer, stomach, liver, and breast. These 6 cancer are very deadly. And that is why, though 150 times chemicals and UV rays, everything. And these chemicals and UV rays, they cause disturbance in the internal genes, internal factors. So together with the external or internal factors, or particularly only with the external internal factors, cancer can be caused. Okay. So what is the good thing, or what is the main important thing we need to focus? uh, WHO is telling that out of this 150 cancers, 1/3 of cancers can be caused by 5 leading lifestyle changes. I'll tell you what are the lifestyle changes, different lifestyle changes. If you feel that if you have any in your, you follow, if you have it, then you just check that, you just be aware that you or me are prone to cancers. Say, for example, what are the 5 lifestyle leading 5 lifestyle changes? If a person is having high body mass index, high BMI, if a person is eating low fruits and vegetable intake, if a person is having a lack of exercise, if a person is a tobacco consumer or alcohol user, in case if someone is falling under these 5 categories, 90% chances are that the person can have cancer in his or her life, right? Okay, so who will get cancer? Many of them will ask the question. They are normal, they are very good, there nothing happened, no familial history, but suddenly cancer happened. How? Because it has become a lifestyle disorder. That is what this. Okay, and let's come, this is about cancer. If you come across, there are 2 different types of cancer treatments. One is conventional cancer treatment, the other one is non-conventional. Conventional cancer treatment is accepted, widely accepted cancer treatment. Non-conventional cancer treatment is not widely accepted because they lack scientific validation. Okay, and mainly what conventional cancer treatment does is that they focus on cancer cells. They want to kill the cancer. That's it. So we can say that chemotherapy is a form of conventional cancer treatment, because the main focus on chemotherapy is killing the cancer cells. But at the same time, the patient will be also be affected because of cancer therapy, right? So they don't focus on patient's well-being, but instead they focus on cancer cells. But non-conventional treatments, they focus on patient's well-being. I think you are getting an idea. Now let us see roughly what are conventional cancer treatments. Before I tell you what is photodynamic therapy, you need to understand what regularly it has been there. Just a background of cancer and its treatment. That's what we are discussing. What are conventional cancer treatments? As I told you, these are basic methods. Okay, and they are regularly used by healthcare professional. Mainly 3 different treatment strategies, no matter whatever the tumor it is there. Mainly 3 treatment strategies or therapeutic strategies will be used by an oncologist. What are the 3 treatment strategies? The first one is surgery, that is surgical removal of cancer cells. Then the second one is radiotherapy, that means with the help of radiation, okay, the cancer cells will be killed, the diseased part will be killed, or the tumor size is reduced. And apart from this, through few medicines can kill cancer cells or control the size of the cancer tumor. So chemotherapy is the third treatment. So regularly, if you get into any cancer hospital, they'll be giving these three different kinds of treatments. So these are called as conventional cancer treatments. They focus on cancer cells, killing the cancer cells, but they do not focus on the patient's well-being, and that is the reason you will be having a lot of side effects because of the treatment. And that is the reason sometimes recurrence. What is recurrence? You think that cancer is completely taken off by surgical removal, but after 5 or 7 years the person may again get cancer, because even one or two cells remain out of millions of cells. It can multiply and it can trigger the cancer again, right? Now, what is the point? What is the non-conventional cancer treatment? Non-conventional cancer treatments are otherwise called as alternative medicine or complementary medicine. Say, for example, nutritional therapy, natural therapy, even meditation, diet and nutrition, naturopathy, homeopathy. All these are called as complementary medicine or alternative treatment or non-conventional treatment, because these things is not scientifically validated most of the time. That means it may work for one person, it may not work for other person. That is why it is called as subjective. Sometimes this alternative treatment work better than conventional treatment. Say, for example, many a times you must have heard about it. A person who has taken, who is affected by cancer, but he has taken medicine from that person, this Ayurvedic doctor, this particular native medicine they have consumed, their cancer, fourth stage cancer, is completely cured. They are
Sorry,
normal.
your voice is not coming here.
Uh, are you getting my voice now? Is it
Yes,
clear?
sir. Yes,
Yes, sir,
yes, sir.
it is clear.
Hello.
Okay.
His voice is very good.
Maybe you can check with your internet connection, or I'll
Durgesh
also.
ji, check with your internet. He's audible.
I think my tower is fine.
No,
We'll
no,
work
you
it
are
out.
audible.
Okay. Yeah. So sometimes this conventional, non-conventional treatments may work better than the conventional treatment, but it cannot be repeated. Say, for example, for one person it has happened very well, and if you repeat the same treatment with another person, it may not happen. Okay, so people have tried a lot, but the repetition or the what to say, the repetition, they are not able to get the better results. So this non-conventional cancer treatment is not widely accepted because it is subjective, right? Okay, now there is a third category of cancer treatment apart from this conventional and non-conventional. There is a third category of cancer treatment. This is where research is playing a role. What is this third category of cancer treatment? It is called as advanced cancer treatment. These advanced cancer treatments are born with the research input or the research contribution on the conventional and non-conventional treatments. We know about conventional, we know about the non-conventional, we know the pros and cons. When people did some modification and people did some research and they made some alteration, and when they found out some good effects, those treatments are called as advanced cancer treatment. What are those advanced cancer treatments? Say, for example, you must have heard about immunotherapy. Okay, treatment of cancer with antibodies or vaccines. Okay, and gene therapy. See, the main problem with cancer is the alteration in the gene, modification in the gene. Why can't we correct the gene? Okay, say, for example, CRISPR-Cas9 gene editing, you must have heard about it, right? That's been used
Yes,
for many. Yeah, so for even for cancer also it can be used, gene therapy. Then the other one is hormonal therapy. Okay, so some of the hormones play a vital role in metabolism, and some of the hormones on and off, they'll be playing a lot of role in the metabolism, and they will be having an effect on cancer. And some of the hormones will be having some receptors particularly which is overexpressed or less expressed in the cancer cells. So treating the cancers with hormones or hormone inhibitors, that is hormone therapy. Then stem cell therapy, it's a very fashionable word, stem cell therapy. That means two different stem cell therapies there. One is our body stem cell can be differentiated into any cells. That is one type. The other type is cancer stem cells. Why this is why cancer recurrence is coming. What is recurrence? A person is healed from cancer, but after 10 years the cancer has come again. That means something, some culprit is hidden there, which caused cancer again. What is that culprit? It's nothing but cancer stem cells. So there are some cancer stem cells which will be underlying, they'll behave like normal cells, but later they'll become cancer cells and they'll come. So killing these cancer stem cells, okay, by stem cells, that is what the stem cell therapy itself is, right? So focusing on cancer stem cells and nullifying them through our body stem cells or modified stem cell or any other agent, that is called as stem cell therapy. The other point is that some stem cells will facilitate the influx of anti-cancer drugs. Say, for example, some cancers cannot be directly treated by the anti-cancer drugs. For a classic example is non-solid tumors. There are two different types of tumors. One is solid tumors and another one is non-solid tumors. Solid tumors are some tumors which you can feel it, you can see it from your naked eyes, you can touch it, you can see the bulging, it is a solid. But non-solid tumors are, it's formed in the fluidic matter. Say for example, it is formed in the lymphatic system, it is formed in the blood circulation. When the blood cells, when the WBCs are affected, lymphoma, okay, all these things are non-solid tumors. Okay, so for that, this stem cell therapy can work really well, right? What are the other advanced cancer treatments? Phytotherapy, nanomedicine, targeted therapy, and photodynamic therapy. All these four items I have already explained you in the beginning, right? So I'm not going very deeper. So these are called as advanced treatments. Why do you call this as advanced treatments? It is the same conventional and non-conventional method only, but these were born because of the birth by the research on the conventional and non-conventional treatments. Right. Let's get our focus into few different things. In the latest realm, if you see, a lot of invention has contributed that immunotherapy worked very well for many different kinds of treatment. Okay, nowadays, how many if you can give me an answer, how many different types of conventional treatment I told you in the beginning? How many different types of conventional treatment? If you have noticed very well, I have told you three different types of conventional treatment: surgery and chemotherapy and radiotherapy. Now immunotherapy has also been added as a conventional treatment because of the research advancement, because of its reproducibility, and because of its efficiency. Nowadays almost all the cancer treatment hospitals, treating hospitals, they are implementing immunotherapy also in their treatment regime. So people will be undergoing, apart from these three conventional, they'll be undergoing immunotherapy as well. And there is a recent discovery which says that immunotherapy can treat almost all cancers. That's what, right? Okay, so these are the effect of advanced cancer treatments. This is of the use of the research in the cancer treatments. This slide, I'll come to it later because I don't want to, focus much onto it, because I wanted to give my focus particularly on photodynamic therapy today. Just I'll give a small introduction or just a small glimpse on this. These advanced cancer treatments, which we were mentioning, right? What they focus on different areas in our body. Okay, they'll be having multi-targeted, they'll be having multimodal effect. Okay, so the advanced cancer treatments, they trigger immune cells, or they may directly contribute DNA damage, and at the same time they'll induce anti-cancer effect, and at the same time they'll have a protection effect on the normal cell, that is dual effect. They also work for the patient survival, which the conventional treatments lacks. Okay, and thereby they bring apoptosis and many other stuff. Okay, right? There is a recent word, ferroptosis is also there, right? Iron linked apoptosis. Maybe one time we'll discuss about it, but now we'll limit it to only photodynamic therapy. Okay, so this is our present focus. And what is a photodynamic therapy? First, a photodynamic therapy is a non-surgical treatment. Okay, and it is a non-invasive treatment. No, invasive treatment means you're harming the patient's body. Okay, you will be having some injections, you will be cutting opening the patient, the pain will be more, or there'll be a pain during the treatment procedure, or it can be done by specialized people, and it may cause some effect. that is invasive. Whereas photodynamic therapy is a non-invasive because it's a non-surgical treatment. Many a times you don't need surgery. Okay. So what is photodynamic therapy? Very simple. Three different components will be involved in it. Okay, three different components will be involved in photodynamic. What are the three different components? If you can see, the first component is light. The other component is one chemical called as photosensitizer. The third component is called as oxygen, which is normally available in all our cells. When these three components are combined, and they were combined in a very perfect form, it will kill the cancer cells by causing damage to us. So when these three joins together, they form oxidative stress, and finally they'll kill the cancer cells. How it will be done, we'll see it. This is a mechanism of photodynamic therapy. Okay, theoretical point of aspect. This is a mechanism of photodynamic therapy. What is happening? This PS is nothing but a photosensitizer. Here you are seeing, I think you are seeing my arrow mark, right? This PS is nothing but the photosensitizer. This is just a chemical compound. Okay, it's a chemical. This photosensitizer is a non-toxic material. Okay, it's actually a non-toxic material. Unlike the other cancers and drugs, it is a non-toxic material. But this photosensitizer, this chemical compound, when it is lit by a light of a specific wavelength, you need to understand there are certain molecules in this earth. They'll be having their unique, almost all the molecules, they'll be having their unique UV spectrum. Okay, they'll be having their unique excitation and emission. Okay, they'll be having their unique spectrum. These photosensitizer or chemicals, when they are lit by a light of a particular wavelength, say for example 620 nanometer or 520 nanometer, 680 nanometer, if they are lit by a particular light of wavelength, what will happen? The particular wavelength of the photosensitizer, okay, is passed. When it is passed, it will absorb the photons, it will absorb the light energy, and it will go to an excited stage. This excited stage is nothing but the photosensitizer molecule has got some energy from light, and it has jumped to the higher level excited state. Now we need to understand this excited state is not permanent, it is very temporary only. So what will happen? From excited state, they'll come back to the normal state. For example, I'll tell you a simple example. You throw ball to the sky. What will happen? You are applying energy to the ball, you're applying force to the ball, and you're throwing it. Now what is happening? It is going up to the sky. Do you think the ball will stay on the sky itself? No. After some time, the ball will come down. It is actually coming down, correct? So likewise, this photosensitizer, when it is absorbing the light, it will go to the excited states, but it is very short time, so it will come back to its normal ground state. When it is coming to the ground state, it is passing through another state called as excited triplet state. Okay. Now this excited triplet stage, when it is coming down, it may lead the light in the form of phosphorescence or fluorescence. You got it? Now what is happening when the photosensitizer is absorbing the energy, and when it is coming to the ground state, it releases the absorbed energy to the cells. The absorbed energy which is released to the cells is picked up by the oxygen in the cells, particularly the oxygen in the cells, and this oxygen is converted, this normal oxygen is converted to a toxic compound that is called as reactive oxygen, which is called as reactive oxygen species. There are a lot of reactive oxygen species. Say, for example, hydroxyl radical, singlet oxygen, O2 dot, you might have seen, right? Singlet oxygen, hydroxyl radical. These are called as reactive oxygen species. These reactive oxygen species, what it will do is that it will go and bind to the different components of cells, particularly to the cell membrane and inside the cell membrane, and they will cause damage. How they cause damage? They'll react to them. Say, for example, you know very well the cell membrane is phospholipid bilayer, and it is a fluidic membrane. When this reactive oxygen species is going and binding with the fluidic membrane, it breaks the membrane. Once the membrane breaks, that's it. The cell is no longer alive. So that is called as oxidative stress. When the reactive oxygen species is more, it causes oxidative stress. When oxidative stress is happening inside the cells, what is happening? The oxidative stress is a signal, triggering signal for the cells to undergo to different pathways. So there is a particular pathway called as oxidative stress mediated apoptosis. So when the oxidative stress is more inside the cells, it will trigger the cells to undergo apoptosis by activating certain genes, and as a result, the cells will undergo apoptosis, or if it is given in higher dose, even they undergo necrosis. But the point is, finally the cell is died. Now, how you are making the cells to die? With the help of light, with the help of light energy, not with very severe toxic compound. So this is the concept behind photodynamic therapy in the treatment of cancer cells. I think you are getting some idea behind this, right? Say for example, I'll just present one simple idea or one simple work that we have done. Say for example, what we done is that we combine this photodynamic therapy along with the nanomedicine. What is nanomedicine? When you're making any medicine or when you're making any compound, and if the compound is in 10 power minus 9 meter, that is very nanometer. When the compound or a molecule, if it is in nanometer size, then it will be having a lot of different effects. Okay, I'll simply explain you how it can be done. Say for example, water, it is having a different effect. If the water is heated and if the water is made into a vapor, the vapor will be able to produce energy in such a manner that even you can run through the vapor, through the steam, you can even run the train. That much energy it can produce. But whereas the water cannot produce, right? But this gaseous form of water can produce that much energy. And if the same water, if it is frozen, it is having a lot of different effects. Exactly in a similar manner, if a molecule is made into a nano level, then it will be having different effects, which can be beneficial and it can be used as a nanomedicine, because it will be having a lot of medical effects. Okay, effects related to medicine, a lot of pharmacological effects will be having. Now I'll tell you one thing. What we have done is we have taken one compound which is called as catechin. We've taken one compound, we've modified this compound catechin. We'll modify this compound catechin and we made that into a nano conjugator. Say for example, what we have done, this catechin is a compound which is present in tea. Okay, this catechin which is present in tea, it's a T5 polyphenol. It's having anti-cancer activities. What we did is that with the help of chitosan, we modified it and we made it into a nanoparticle through some methods. And this nanoparticle is targeted along with folic acid. This nanoparticle is targeted along with folic acid. We attached a ligand called as folic acid and we attached it. Now this is what you call it as nano engineering. This is what you call it as nano engineering. Now what will happen? Why we are attaching folic acid? Because the cancer cells will, some cancer cells will be having lot of folic acid receptors on its cell membrane. So not other normal cells will not be having the folic acid receptors more, but the cancer cells will be having a lot of folic acid receptors in the cell membrane. Now what will happen when you make a nanomedicine like this? If you are making a nano conjugate like this folic acid will go and bind with the folic acid receptors, and wherever the folic acid receptors are there, this compound will go and bind there. Okay, so it will spare the normal cell. More amount of this chemical will go and bind to the drug, will go and bind to the cancer cells. Now what will happen? Already this is a nano formulation. When a drug is in nano formulation, it will be having lot of effects. It will have increased surface area. It will have a sustained release. That means, say for example, your doctor is asking you to take paracetamol every 4 hours, right? If you're having continuous fever, morning you have to take a paracetamol. After 4 hours, afternoon, till afternoon you won't have fever, but again afternoon you will be having a fever. So again you have to take paracetamol. Then it will be effective only for 4 hours. Then evening you have to take again one paracetamol, correct? Now, what is sustained release? Imagine if we make this paracetamol into nano formulation and it is having an effect called a sustained release. That means if the paracetamol is able to release the paracetamol, the tablet is able to release the paracetamol slowly, whatever your body needs. you can make in such a way that one tablet is enough for the entire day. So one tablet 500 mg or 650 mg, slowly it can release whatever your body needs. So for 24 hours, we can make it to release even one tablet. What is happening here? The extra doses of the drug is being reduced. So the side effect is being reduced. Likewise, lot of properties this nanoparticle will be having when you make a drug into nanoparticle. So this triple conjugate we are giving to the lung cancer or breast cancer. We have tried with many different cancers. Lung cancer also we tried, breast cancer also we worked out. Now what is happening, this folic acid is going and binding to the breast cancer receptors or lung cancer receptors, and they release this catechin. Apart from this catechin, we are also conjugating this catechin along with a photosensitizer. We found out when we made modifications in the catechin in the normal state, it is not a photosensitizer. But in case if we do some modifications in the catechin, it is becoming a photosensitizer. Photosensitization means a compound which can absorb the light energy when a specific wavelength of light is being to it. Now, what is happening? Now we made this catechin into a photosensitizer by modification, and we tagged it along with another photosensitizer called as zinc phthalocyanine. Okay, together with these two, when we insert it to a patient or when we insert it to cancer cells, and when we give light of a particular wavelength, we found that the apoptosis being triggered and the cancer cell death was being born. Right? Okay. Say for example, there are few other works which we have done related to this. I have showed you only about catechin, but we worked on different items like the different polyphenols like ferric acid, gallic acid, and turmeric conjugates. So many different modified compounds, we made them into photosensitizer. For you to make some understanding, this photosensitizer is a chemical compound. There are a lot of chemical photosensitizer are available, but now due to research, we have researched, and so many researchers have researched, and they found out many natural compounds are having this photosensitizing activity. Say for example, hypericin is a wonderful photosensitizer which is extracted from, which is an active compound from Hypericum perforatum. Okay, so this compound is from plant source. Now they are using this hypericin as a FDA approved drug for photodynamic therapy. Right? You can ask me in the question answer session where it is being done, how it is done, where in India. I'll talk to you about all those stuff. But let's try to focus on the work here. Now, if you can see few works what we have done, I told you about reactive oxygen species, right? Now, if you see the photodynamic therapy or the combination of this drug, what they have done is that no, they have increased the reactive oxygen species inside the cancer cells, and this is the result. You can see that when they are treated with the the combination of the drugs, or when they are treated with the light and the cancer drugs, and along with the photosensitizer, what is happening in this nano conjugate or the nano formulation of this drug, they're increasing the reactive oxygen species. You can see by the increased fluorescence, the control cells are dark, but when the reactive oxygen species are increasing, you can see the light emission is being increased. You can observe this. Okay, so these are initial stages, or this is an indication that cancer cells, because of treatment, they are undergoing increased reactive oxygen species or oxidative stress. Okay, now there's one more thing that we can see, that the formation of apoptotic bodies, or whether the cells are undergoing apoptosis. You can see from these two cells, it's done on cervical cancer cells. Forget about all the agents whichever we have used. I'm showing you different treatment methods or different results which we have done in the past few works, and for you to understand that how the photodynamic therapy and nanomedicine can bring about a significant cancer cell death and significant contribution to the cell therapy or cancer therapy. Here we can see the control is rated in the green fluorescence. But if the cells are undergoing apoptosis, you can see a mix of green and red and orange fluorescence. So when the intensity of light is shifting from green to orange or deep red, you can say that the cells are undergoing apoptosis more. So more the red or orange fluorescence, more number of apoptosis you can see. And even you can see significantly visually, you can see the apoptotic bodies which are formed here. Okay, 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 fine. Now, this is one thing which we have done it on the rats in the rat model. Okay, this we have done it on the colon cancer. So now what is happening here? Even photodynamic therapy can be used for external cancers like skin cancers and oral cancer. But at the same time, cancer therapies can be, photodynamic therapy can be used for internal cancer as well. Okay, so the light has to be, passed, okay, inside, and it can be lit in a particular, area. And there are certain drawbacks of this photodynamic therapy as well, because it cannot go very deeper into it. But still, with the advancements in technology, the penetration of light can be deepened. Now if you see this result, the first two pictures, it shows that how the normal colon architecture is. You see a small dot, okay, you see the normal colon. But if you see the picture of C, D, and E and F, you can see here and there the colon is not, it is being bulged. It's called as ACF. It's nothing but aberrant crypt foci. Okay, aberrant crypt foci. Polyps are being formed. This polyps will be later. Okay, these polyps will be later. Okay, will be formed into the cancer. If you see the C, it's the C and D. If you can see that the cancer is being formed. It's not a normal colon, it's an aberrant crypt colon. Now colon cancer is being formed. The next stage is colon cancer. But if you see, when they're treated with the light, when they're treated with the drug, when they're treated with the nanoconjugates, different other works, what is happening? The number of aberrant crypts and the size of the aberrant crypt is being reduced. That you can see it here. And similarly, how it can bring about the immunomodulation. Okay, we did some work on the bone marrow cells, and we found how the protective effect is being carried out, is being seen. One more thing I'll show you. This is a typical evidence on DNA damage. Okay, typical evident how DNA damage is. This is a picture of the comet assay. What you understand is a comet assay. Comet assay is nothing but, in case if the first two cells in the control, you can see the cells are fluoresced, the cells are lightened like a dot. Okay, it's a perfect round. But when the cells are damaged, when the DNA is damaged, when you run the electrophoresis, what will happen? The DNA will be separated because of its different, a length of the fragments. And when you fuse it with the fluorescent dye, you will see the appearance of a comet. You know what is a comet, right? So the more the damage, more the tail of the comet will be there. You see, less the damage, less the tail of comet is there. But if the damage is more, then the tail of comet will be found more. Then you can see the tail of comet is more. So as the treatment is being increased because of the nano formulation and because of the light and many other stuff, because of PDT, photodynamic therapy, what is happening here? The cancer cells DNA damage is being increased, and they show a pattern of DNA damage. This is a qualitative result, but we have the quantitative aspect result as well. We have calculated the tail length, we have calculated the tail moment, olive tail moment, and everything, and we justified that. We found out that yes, quantitatively it is bringing a DNA damage. Okay, the treatment is bringing a damage. And here you can see how the in the lymphoma, how you can see how the size of the solid tumor is being reduced because of the treatment, right? How the size of the solid tumor is being reduced because of the anti-cancer treatment and because of the light and everything, right? And these are the morphological changes. What observed is, the survival rate. So the cancer induced animals were dead, mean survival time is around 18 days, 18 to 20 days. But in case if they are given with the treatment, the animals were found to be living more than 46 days at an average. After that, we stopped the experiment. But at an average, even after 45 or 46 days, the animal was surviving, right? And these are some molecular analysis how it's being done, right? Okay, fine. So this is the rough idea about uh, uh, the results and the photodynamic therapy. Generally in India, what is happening? Generally in India or all over the world, photodynamic therapy. Earlier it was not practiced, but now it is widely practiced. Now it is practiced. Earlier it is only for the superficial cancers, but now they're even practicing. It has gone up to clinical trials as well, and many clinical trials have proven results, particularly when it is done, when the photodynamic therapy is done. And many a times, even photodynamic therapy has proven to treat even the patients with advanced cancer stages, even when other cancer treatments failed. But this nanomedicine and photodynamic therapy combination, or combination of this photodynamic therapy and chemotherapy, many other stuff, photo radiotherapy and nanomedicine, they brought about a significant treatment effect on the cancer patients, which have been proven on the cancer patients, not just on the preclinical models. So this is a rough idea about what is nanomedicine and what is a photodynamic therapy, and how when these are clubbed. For a few reasons, I couldn't show you the exact results of some of the nanoparticles, their spectrum, their TEM and SEM analysis, and also other molecular capabilities of this photodynamic therapy, because I was told not to show few important things which we are planning to have it for publication or for patents and everything. For copyright reasons, we are not showing that. But otherwise, this is what, for the couple of years from 2019, we have seen that the tremendous effect on photodynamic therapy through our research, through different modifications, it has been brought in our preclinical models as well, right? So what is the overall positive message? According to WHO, 30 to 50 percentage of cancers can be preventable. Okay, the first point is prevention. Prevention is better than cure. We are coming to the end of this. So the first take home message is 30 to 50 percentage of cancers can be preventable. And if the cancers can be screened early, most of the six cancers which I told you earlier, which is considered to be very lethal, they all can be screened. So if the cancers can be screened earlier, more than 60 percentage of tumors can be detected earlier. And if you see how cancer screening can happen, the government has its own modus operandi all over India to have cancer screening, okay, which is happening everywhere. Like how polio drive is there, this cancer screening is happening every now and then. But because of social stigma, sometimes people are not preferring it, particularly when it comes in terms of women. When it comes to breast examination or cervical examination, people will not come forward, for social stigma and everything. But we need to break all the odds, and if you can see, if you go further for the screening, more than 60 percentage of tumors are preventable, right? And what is the other important thing? I told you, not I told, I just told whatever the WHO said. If we bring about lifestyle changes, 55 major cancers or six major cancers can be reduced. Okay. So if we adopt those positive lifestyle changes, okay, it can bring a reduction in the risk of five major cancers, which is being branded lethal. And diet, even they've told that diet and exercise, they're consistent enough, we can prevent 30 percentage of the cancers. And reducing or quitting smoking helps to stay away from cancers. So prevention is better than cure. So this is a positive message. Most of it, I just want to add one important thing is that my most of my research is done on the dietary phytochemicals. Right from 2007, I was been extensively, I'm working on the dietary phytochemicals, particularly on polyphenols, phenols, and many other ingestible substances which is found in our diet. I'll tell you one clear fact that whatever we have taken almost 35 to almost 50 different dietary substances, and we have tested its effect against cancer, not single cancer, different cancers. We did some modifications. We made some nano, conjugates out of it, and we worked. And I have to tell that out of everything, all the 100% of modifications on the native dietary phytochemicals have significantly shown to have anti-cancer effect. And we should be proud that we all are having those in our diet, right? So together with that and this, with the proper lifestyle, I think we can beat cancer. We can prevent it before it comes. Even if it comes, early cancer screening and an early diagnosis and treatment can be very well effective for a patient to win over the cancer. That is what the positive message is, right? So now it can come to any person, right, from children to age old person. So a little awareness on cancer is needed, and little awareness on the treatment strategies is needed. And if that is clubbed together with a positive lifestyle and mindset, I think we can beat this a monster called as cancer. So it's preventable and it is treatable, so you can be free, right? I think most of your students and most of your researchers who are attending, so if this helped me, these are 8 different ideas which help me. Just have a passion, work for the passion, and focus on your passion. Push forward. Sometimes we fail, sometimes we fail, but no matter, stand up and push forward, and recreate different ideas, improvise, and most importantly, help each other. Okay, help each other and persistent enough, persuade towards us and be persistent. If you are doing like this, definitely one day you'll be winning it, and definitely success will be yours. I wish you all the very best, and I want to give my acknowledgements to all the funding agencies and different bodies who helped our research in different aspects. So all of the research contributions are due to their support. So I would like to thank PDU and Department of Biotechnology, School of Energy Technology. Apart from all these funding agencies, I thank.
Thank you very much.